Hi everyone. This is the first lecture of SCB 204, Human Anatomy and Physiology Part 2, Section 732. We're going to meet on Mondays and Wednesdays from 1 o'clock to about 4:30. About 4:30. I'm your instructor. I'm Professor Howard Motoike. Okay, here's my office, but I won't be there. Okay, here's my email. I try my best to answer all emails as quickly as I can. Okay, here's my office phone number, but I won't answer it. So your best option is to email me. Okay, and my office hours will be before class, usually. Okay, and I'll send you the link for each class. Now, SCB 204 is pretty much set up much like SCB 203. You have 6 quizzes where we drop the lowest, and you have a midterm and a departmental final exam for about 30%, which is the lecture part. The laboratory part has 4 exams with 40%, and that gives you 100% for the entire course. I'll let Professor Kullaway, talk more about the laboratory breakdown. Okay. The lecture quizzes are multiple choice and true and false. There'll be 25 questions and you'll have 30 minutes. The midterm and the final will have 50 questions as well as true and false, and you'll have an hour to take the test. Here are the quiz and exam dates. I think I've got this correct. We have to do some modifications to the syllabus for the short semester. So the actual material covered for each quiz will be modified so that we can fit all the quizzes into the semester. Keep in mind that we have 6 quizzes and a midterm we have to fit in 6 weeks. And the laboratory portion is heavier near the end of the semester. So I tried to avoid having 2 quizzes in the latter part of the semester, and so I tried to put quizzes earlier. Okay. I think this is still current. If you have the code for mastering A&P, here's my course ID. Okay. If we find that it's no longer works, I create another one. If you bought the book from the bookstore and you have the code, okay, or you received a free code, it might still work. Okay. And and the instructions will be on Blackboard. Okay. I have. At the time of me recording this, I haven't done it yet because I have to find it somewhere buried in my files. Okay, but it's not a requirement for the course. Okay, this is all voluntary to use Mastering A&P. Here are the textbooks that we use. Here's the laboratory manual, and here's the lecture text. Okay. The laboratory text, muscles, I don't know how much you're going to, you probably used it maybe for 203 if you took it prior to spring 1. And the dissection might be helpful too for this semester for SCB 204. Okay. So here's this is the first lecture on SCB 204. Oh, one thing I should note is that unlike my SCB 203 lectures, which are all kind of the same length, my SCB 204 lectures are all different lengths. Generally, they're shorter at the beginning of the semester, and we get longer towards the organ systems near the end. So just because I finish a lecture, if there's time remaining, I'm just going to go to the next lecture, because at some point, I'm going to need the time for the longer lectures later in the semester. So I'm just letting you know that many of these early lectures are very short. Okay. So this introductory lecture is on the histology and organization of the nervous system. Okay. And so in most A&P courses, you do this in the first semester. Okay. Well, unfortunately, before I got hired, the nursing department wanted certain things to be taught in the first semester because that's what they required at the time for the nursing program. And so we just never changed everything back. So we're probably different from the rest of the world by having the nervous system in the second half of the course. Okay, so histology and organization of the nervous system. Now, much more histology will be covered in the lab, so I'm not going to go too heavily on it. Okay, and so here are the objectives for today. We're going to talk about the functions and the divisions of the nervous system. We'll talk maybe of parts of the neuron and the structure and function of some of the different neurons. And we're going to talk about a unique cell type called Neuroglia cells. Okay, and we'll talk a little bit about myelination of the central nervous system and the peripheral nervous system. And then we'll introduce some terms that you're going to have to remember for the rest of the semester and beyond. Things like nerve, tract, ganglion, and nucleus. And I think this last step I removed last semester. I just forgot to update the objectives. Okay, so let's begin. What are the major functions of the nervous system? Well, they're going to control perceptions and experiences. So everything you see, everything you do, all your life experiences, these are all going to be recorded by your nervous system. Okay. So perceptions, anything you touch, you feel, you see, you hear, these are all going to be parts of your nervous system. Directing voluntary movement. Okay. So for example, your skeletal muscle is primarily voluntary. So if you want to contract your biceps, you can. If you want to stick your tongue out, you can. Okay? Because it's voluntary. The nervous system also functions consciousness, personality, learning, and memory. Okay? So you're reading this because of your nervous system. Okay? You like it or you don't like it, what I'm saying, because of your personality. Okay? And you're aware, hopefully, of your surroundings, of your consciousness. Okay? And if you can remember these first three, then you have memory, which is also part of your nervous system. Okay. Finally, the nervous system regulates many aspects of homeostasis. Okay, this was the magical word you learned in SCB 203. Okay, and if you remember what it means, it's the maintenance of a constant internal environment. Remember, your body likes same old, same old. It doesn't like change, and so it's going to keep things constant if it can. And it does this also with the endocrine system. Okay. So the first half of the semester is going to be on the nervous system and the endocrine system. And then we go to the other organ systems that are remaining. So let's look at the overview of the nervous system and look at its anatomical divisions. Because remember, this course is both anatomy and physiology. And you learned on the day one of SCB 203 that anatomy is structure and physiology is function. So it's even more evident in the nervous system where we have both structural and functional aspects. Okay, so let's look at the anatomical divisions first of the nervous system. The nervous system is divided into two parts. That's nice. I like things that are divided into two parts because it's simple then. You have what's called the central nervous system, or the CNS, and the peripheral nervous system, or the PNS. Okay, so the nervous system is divided into these two sections: the central nervous system and the peripheral nervous system. Okay, so what's composed of the central nervous system? Well, again, there's only two parts to it, which is nice, makes it simple. The central nervous system is composed of brain, okay, that's in your cranium, and your spinal cord that extends inferiorly down into the vertebral canal of your vertebrae. Okay. Here we show an illustration. That's your brain, okay, and here's your spinal cord. Together they make up the central nervous system, okay. Now, what makes up the peripheral nervous system? Okay, peripheral means they're in the periphery, okay, so they're more tangential to the central nervous system. And this is even easier because it's only composed of one thing. They're composed of nerves. Okay, I guess I didn't need the 1 here, but nerves. Okay, and these are like little highways to different parts of the body that carry signals to and from the central nervous system. So they're two-way streets. Okay, so here's your peripheral nervous system. Okay, and they're composed of nerves. Okay, later on we'll mention about the different types of nerves. We have spinal nerves and we also have cranial nerves. But for now, the peripheral nervous system is composed of nerves. Okay. Now that we told you that the peripheral nervous system is composed of nerves, what is a nerve? Okay. Well, a nerve is a kind of a potpourri of different structures that include bundles of axons. Okay. Of course, we haven't told you what axons are yet. These are these long extensions of neurons that are packed with blood vessels and connective tissue. So this package is what's called, or are collectively called, nerves. Okay. And so now I tell you that there are different types of nerves. We have cranial nerves, okay, that enter and exit from the brain, and there are also spinal nerves that exit from the spinal cord. Okay. So now we could go back to our figure, and now we can say, there are different types of nerves: cranial nerves, and we have spinal nerves. Okay, you can see the cranial nerves are coming from the brain, or to and from the brain, and the spinal nerves are coming to and from the spinal cord. Okay. Let's talk about the cranial nerves. We'll talk more about them in a later lecture, but we'll introduce them now. Okay. There are 12 cranial nerves that originate from or travel to the brain. Okay. And each of these 12 cranial nerves have a name, and they also have a number by a Roman numeral. And eventually you're going to have to learn them by name, number, function, as well as their origin. So if you want to begin studying those, you might want to do this because you're going to have to know it both for the lecture and for the lab. So it's going to be a double whammy. But you learn it once, you're already set for both parts of the course. Okay. Of course, in the lab, you're going to have to identify them. Okay. You don't have to identify them here in the lecture. Now, spinal nerves, you have 31 pairs of spinal nerves that originate from or travel the spinal cord. What an odd number, 31. Okay. And we'll mention why there's an odd number in a bit. Okay, it seems a little odd number. Okay, and in the lecture you might learn that as well too. I mean in the laboratory. Okay. Okay. So let's look at the functional divisions of the central nervous system, and they're based on 3 general functions. Okay. So if you had to divide functional divisions of the nervous system, you end up with these 3. Okay, they're divided into sensory functions. Okay, so it allows your body to sense things, touch changes. There's integrative functions. It decides what to do, and there are also motor functions that causes, for example, muscles to contract or maybe glands to secrete, for example. So these are the 3 functional regions of the nervous system. Okay, so I made this little handy dandy table for you. Okay, so here are the 3 major regions of the nervous system: sensory, integrative, and motor. And we see that sensory is part of the peripheral nervous system. And it has another name, a structural name called afferent. Okay, because generally these are going to a structure and collects information about the body. To the nervous system. Integrative is mostly in the brain, and this analyzes and interprets information in preparation for an appropriate response. So it's sort of like when you're saying, you're thinking about it. But here it's your nervous system deciding what to do based on the sensory information. Okay. Finally, we have motor functions, and this is primarily in the peripheral nervous system, and these are efferent because they're going away from the brain or the nervous system, and these are actions in response to integration. Okay, and so here we see this girl kicking a soccer ball. And this one simple activity includes all functional aspects of the nervous system. Okay, so for example, there's sensory input. Okay, so she sees the soccer ball, and she sees the soccer ball coming toward her, and these sensory signals center her brain. So she has to decide she wants to kick it, but she has to make sure she kicks the ball at the right moment. So this requires what's called integration. So her brain receives and integrates the sensory input and sends signals for an appropriate motor response. And so that travels down the spinal cord and out to the leg muscles here. And the muscles of her thigh and leg receive the motor signals. They contract and kick the soccer ball with perfect precision. So this illustration illustrates the three major divisions of the nervous system: sensory, integration, and motor input. Okay, so let's look at the functional divisions of the peripheral nervous system. Okay, so here, this is a figure from your textbook, and I removed some of the words that we were going to fill in. Okay, I left a few things because we learned them already. We have the central nervous system, and we have the peripheral nervous system. And we know the peripheral nervous system includes cranial and spinal nerves, and we know that the peripheral nervous system is a two-way street communicating with the central nervous system. So now we're going to fill in the rest of the table. Okay, so the peripheral nervous system is divided into the sensory or afferent division, because it's taking sensory information and taking it to the central nervous system, which is afferent, and it also has the motor division, which is efferent, which is going to take signals away from the central nervous system. So remember, afferent is going to, efferent is going away. Okay, so now we can fill in these two first boxes here. Okay, so arbitrarily I put the sensory afferent division in the blue one, and I put the motor efferent division in the red one. Okay, we know. I guess it wasn't really arbitrary, but we see the arrows going toward here, so we know this is going to. Here's the sensory efferent because it's taking information to the central nervous system, whereas the efferent division is going away from the central nervous system. So they weren't arbitrarily. I had to put the sensory in the blue one and the motor in the red one. Okay, let's take a closer look at the sensory afferent division. Afferent means carrying toward. Okay, remember we said it's going toward the central nervous system, and that's what afferent means. So sensory is a functional term. Afferent is a structural term. Okay, so information is detected by sensory receptors and taken to the central nervous system by the cranial and spinal nerves. Okay, so these are the peripheral nervous system. Okay, now the sensory afferent division is further subdivided into the somatic sensory division and the visceral sensory division. Okay, so they're both part of the sensory division, but one is somatic and one is visceral. Okay, so here we'll fill these in. Okay, we made the big blue box, the somatic sensory division, and we made the smaller one, the visceral sensory division. Okay, and these are both parts of the sensory afferent division. Okay, so let's look at the somatic sensory division. Somatic comes from soma, which means body. Okay, so these are the sensory division from the body. Okay, so where are these signals coming from? Well, neurons carry signals from skeletal muscles, bones, joints, and the skin are part of the somatic sensory division. And it also includes special sensory neurons for vision, taste, smell, and balance. Okay, so these are all special signals that are part of the somatic sensory division. What about the visceral sensory division? Visceral comes from the word viscera, which is your organs. Okay. So neurons are from signals from organs such as the heart, lungs, intestines, kidneys, and the urinary bladder. They're all examples of the visceral sensory division. Okay. Now let's look at the motor efferent division. Okay. Remember, efferent is carrying away. It's going away from the central nervous system. Okay. So remember we put this in earlier in this red box, the motor efferent division. So now we're going to see, we can see now that it's going to be in two components as well. We have what's called the motor efferent division. Okay. So motor neurons carry two effectors such as muscle contraction or glandular secretion. Okay, so glands and skeletal muscles. And it's divided into the somatic motor division and the visceral motor division. Okay. So here we put the somatic motor division first. We'll leave the last one because it requires a little bit more explanation. Okay. The somatic motor division is also known as the voluntary motor division. Okay, so we have conscious control over it. Okay. And the somatic motor division are neurons that transmit signals to skeletal muscles. Okay. So this allows us to move our muscles like your leg muscles and your arm muscle. Now the visceral motor division is also known as the involuntary motor division. Of course, we don't really use the visceral motor division name too much, and we don't really use the involuntary motor division too much either. But what we do use are this term, the autonomic nervous system. Okay, but you should know that the three are the same: the visceral motor division, the involuntary motor division, or the autonomic nervous system are all the same. And so now we put in our last box. So here's the autonomic nervous system. Now the visceral motor division has neurons that carry signals primarily to the thoracic and abdominal viscera. Okay, and so it regulates secretion from certain glands, smooth muscle, and heart contraction. So these are all involuntary functions. Okay. So let's look at a summary now, the structural and functional divisions. Okay. So now here's from the textbook. This is everything printed together here, and I pretty much have the same information in the slides. So this has nothing new. It has no new information. Okay. So of course, for the quiz, make sure you know how the nervous system is structurally and functionally broken down. Okay, so now if you're a visual person, here this figure shows the same information but with pictures. Okay, so this if you're not a visual learner, well then go back to the slides. But if you're a visual learner, now you can see the actual organs that are involved in the different divisions of the central nervous system. Okay, so far so good. Now, in the laboratory you're going to learn more histology, but we'll do the basics here. Okay, and so some of this you're going to hear again probably on your first lab meeting. Okay, so the nervous system histology, as you might expect, contains mostly nervous tissue. Okay, but it also includes other tissue too as well, like epithelial and connective tissue. Okay, so probably the one tissue type that's not probably present is muscular tissue, too much. And so here's a picture showing the most well-known cell type are the neurons. But notice there's unique cells called neuroglia that outnumber the neuron. Okay, so actually the major cell type are neuroglial cells, and you can see that there's a large extracellular matrix that's unique to nervous tissue and different from connective tissue. Okay, so nervous tissue is composed of neurons and many neuroglial cells. Okay, neurons get all the glory. Neuroglial cells do a lot of the support work. Okay, let's take a look at neurons first. Neurons, like muscle, are excitable cells, meaning that when they're stimulated, they do wonderful things like producing action potential. Okay, so you remember action potential? Neurons generally have three parts: the cell body, where the nucleus is; dendrites, which are small little branches that provide information to the cell body; and an axon, a long extension that sends information away from the cell body. Okay, so we have a cell body, dendrites, and the axon. Okay, let's look at the cell body first, or the soma. Okay, and this is the most metabolically active part of the neuron. Because where the nucleus is, this is where a lot of the protein synthesis occurs. And just like regular cells, it has free ribosomes as well as rough endoplasmic reticulum. But here, they appear densely staining, and we have a special name for them as Nissl bodies. So Nissl bodies are the free ribosomes and rough endoplasmic reticulum of a neuron. And of course, since they're highly metabolically active, they have many mitochondria. Okay, so here's the cell body. Okay, that's the nucleus. Okay, here's mitochondria. Okay, here's your Nissl bodies, ribosomes, and rough ER. It contains cytoskelet elements such as intermediate filaments to help maintain the shape of the cell body. Okay. It also has neural fibrils that support the dendrites and axons to maintain their shape. So here we have little pictures of neural fibrils here. Here are the intermediate filaments. Okay. Of course, just like other cells, they have the organelles you'd find in any typical cell, such as Golgi, nucleoli, microtubules that provide support and a way to transport chemicals throughout the neuron. Okay, let's look at the other part of this a neuron, the dendrites and axons, which are processes that connect to the cell body. And so this is the dendrites and axons are the structures which allow a neuron to communicate with other cells. Okay, so let's look at the dendrites first. Okay, these are short forked processes resembling tree branches. Okay, and they receive electrical input from other neurons. So they send information to the cell body from other cells. So here are the dendrites. These are all dendrites. Okay. Axons is what we typically call a nerve fiber, generate and conduct action potentials away from the cell body. Okay, so here's the axon as it extends, and they can also branch too. These are called Axon Collaterals, but generally, many of them have a single axon. Now, a special area is called the Axon Hillock, and this gives rise to the axon from the cell body. Okay, so here you see the Axon Hillock that extends the axon from the cell body. Okay, and as I mentioned, that you could have branches called Axon Collaterals. And so here I've labeled them here, Axon Collaterals. And on these axons, we have extensions which are called Telodendria or axon terminals. There are many different names for these structures. Okay, so the axons and axon terminals split into what's called telodendria. The telodendria terminate to axon terminals or synaptic knobs that communicate with target cells. Okay, I guess I don't need this term here. This probably could be eliminated, I guess. Because telodendria terminate into axon terminals. Okay. So here are axon terminals and telodendria. Let's talk a little bit more about axons. Okay. They're surrounded by a plasma membrane called the axolemma. Okay. It has cytoplasm called the axoplasm, but they do not contain ribosomes or Golgi bodies. That means all protein synthesis and secretion occurs from the cell body and are transported down the axons. Okay, so here are these terms here. The axonema is a plasma membrane, and the axoplasm is the cytoplasm. So as we mentioned, it doesn't have any Golgi or protein synthesis, so we have to have what's called axonal transport, where things are moved from the cell body to and from the axon. And this can be either retrograde or antigrade. Retrograde is toward the cell body. Okay. Antigrade transport is away from the cell body. Okay. So if something's made in the cell body, it's going to go through antigrade axon transport to the axon. Some things may be brought back, maybe to be degraded perhaps, and is retrograde toward the cell body. Okay, so make sure you know the difference between retrograde and antigrade. Now that we know the parts of a neuron, now we can look at the functional regions of the neuron. And there are three main parts: receptive, conducting, and secretory. Okay, and in this figure here, we took in just a simple neuron, and we divided it into different functional regions. We have the receptive region, we have the conducting region, and the secretory region. And here I made you a little handy dandy table showing the different functional regions, the structural regions that compose these functional regions, and their functions. So the dendrites in cell body are important for our receptive region, and because they receive signals. The axon are the conducting region because they are able to propagate signals like action potentials. And finally, the axon terminals, we're going to find out, is secretory because they're able to secrete chemicals called neurotransmitters. Okay, so make sure you understand and know this table. Okay, now there are many different types of neurons, and we're not going to plan to name each one of them, but we're going to just classify some of them, the major ones. We have multipolar, bipolar, and pseudounipolar neurons. And let's discuss some of these. Multipolar neurons are probably the ones you're probably most familiar seeing, because these are more than 99% of the neurons, and there's considerable variability between them as well. Generally, they have a single axon and multi-branch dendrites. So examples of multipolar neurons are the spinal motor neurons, pyramidal neurons, and Purkinje neurons, or Purkinje cells. Okay, and of course this is named after a person, so it's capital P. Okay, so here in this figure we have the multipolar neurons and three examples: the spinal motor neuron, the pyramidal neuron, and Purkinje cell. Okay, bipolar neurons have two processes, one axon and one dendrite. Okay, and most of these are sensory neurons, such as in the eye and the olfactory epithelium. Okay, so here's a bipolar neuron, and it has a major dendrite and a major axon. Pseudounipolar neurons develop as bipolar neurons, but the process is fused to form a single axon. Okay, the peripheral axon brings sensory information to and a central axon away from the cell body. And these you're going to find are neurons for touch, pressure, and pain. Okay, so you can see they kind of shifted over. So now you have the peripheral axon that's sending information to the cell body, and a central axon that's taking information away from the cell body. So here's an example of a pseudounipolar neuron. Okay. We have different functional classifications of neurons, and we have three classes based on the direction and information the information is carried. And these terms we've actually talked about already. Okay. We have sensory neurons, we have interneurons, and motor neurons. Okay. The sensory neurons we've been calling afferent neurons because they go toward the central nervous system. Okay. Interneurons are also known as association neurons. Okay. So their major job is for integration. And we also have motor neurons or efferent neurons because these are going away from the central nervous system. Okay, so here's another handy dandy table. So we have the functional neurons. I just forgot the N right here. The sensory or afferent, interneurons or association neurons, and motor afferent. As you can see, most of the neurons are interneurons or association neurons. Okay, and the direction that they transmit their impulses, sensory afferent, go toward the CNS. The interneurons or association neurons, they relay information between the central nervous system and peripheral nervous system. And the motor or efferent neurons send the information away from the central nervous system. What is the purpose? Well, the sensory neurons have sensory receptors to the brain or spinal cord. Interneurons are between sensory and motor neurons, and motor or efferent neurons extend to muscles and glands. And here in the last column, I've given you the major structural neuron present for each of these functional neurons. So sensory or afferent neurons are usually bipolar or pseudounipolar. Most interneurons or association neurons are multipolar neurons, and most motor or afferent neurons are also multipolar neurons. So you can see most of the neurons are multipolar. Okay. Now we're going to introduce some common terms you're going to have to learn and know for the rest of the semester. These are what we call neuronal components, and these include nuclei, ganglia, tracts, and nerves. So for now, we're just going to introduce the term. So here I made you another handy dandy table, and you can see the nuclei are clusters of cell bodies in the central nervous system. Ganglia are clusters of cell bodies in the peripheral nervous system. Okay, so make sure you know the difference between a nuclei and a ganglia. Tracts are bundled axons in the central nervous system, whereas nerves are bundled axons in the peripheral nervous system. Okay, so these four terms we're going to be using throughout the rest of the semester. Okay, finally, we mentioned that neurons get all the glory because they do all the wonderful things of our nervous system, but the majority of nervous tissue are composed of a specialized cells called the Neuroglia cells. Okay. And the Neuroglia cells are 6 different cell types found in the central nervous system and the peripheral nervous system. And their job is to help take care of neurons and make sure that the nervous system is a well conditioned place for the neurons to function and do their work. And so they're very versatile, and they have probably many different functions, such as maintaining the environment, protection, and assisting in proper function of neurons. Okay, and unlike neurons, neuroglia retain the ability to divide. Okay, and so here are the 6 neuroglia cells. We have 4 in the central nervous system, including astrocytes, oligodendrocytes, microglia, and Ependymal cells. And the remaining 2 are in the peripheral nervous system. These are Schwann cells and satellite cells, and these make up the 6 neuroglia of the nervous system. So let's go through each one now. Let's start with astrocytes. Okay, so here's a picture of an astrocyte. So it looks like kind of like a star. Okay, and here's another one here with the nucleus in the center. They're the numerous and largest of the neuroglia cells in the central nervous system. And they're unique because they have processes that terminate and end feet. Okay, so here are the processes, and you can see the enlargement at the end. These are the end feet of astrocytes. Okay, so what are the astrocyte functions? They have a number of different functions. They anchor neurons and blood vessels, okay, with their little end feet. They help regulate the extracellular environment. They assist in the formation of the blood-brain barrier. Okay, well, we haven't mentioned this yet, but we will eventually have a discussion on the blood-brain barrier where the vascular system is separate from the nervous system. And they're important for repairing damaged brain tissue. So here's another picture of an astrocyte. You can see that little end feet can associate with neurons and other cell types. Oligodendrocytes are fewer and smaller and have smaller processes than astrocytes. Now, they have an important job of wrapping multiple axons to produce a myelin sheath. Okay, and we're going to eventually see that the myelin sheath is going to allow these neurons to propagate action potentials much faster than those that do not have a myelin sheath. Okay, so here's an oligodendrocyte here in blue. Okay. And what one major note we want to make about the oligodendrocytes is they have the ability to form the myelin sheath over multiple neurons. Okay. The microglia, as you might expect, are smaller and are smallest and the least numerous of the neuroglia. These are unique because they're phagocytes, and after an injury, they ingest pathogens, dead neurons, and cellular debris. So they operate much like macrophages, but they're called microglia. Okay, so here's a microglia cells, kind of star-shaped like too as well. And so here's a microglia cell showing phagocytizing a particle right here. Ependymal cells. These are ciliated cells that line brain cavities and the spinal cord and filled with cerebral spinal fluid. So our central nervous system, it has passages that contain the cerebral spinal fluid, and also it's surrounding the central nervous system. And so it's buoyant in this fluid. And so it's the ependymal cells that produce, circulate, and monitor the composition of the cerebral spinal fluid. Okay, so here you can see the ependymal cells. They're lining a cavity where they're going to be secreting the cerebral spinal fluid. And you can see they have little cilia. So as it beats, it can circulate the cerebral spinal fluid. So those are the neuroglia of the central nervous system. Okay, the neuroglia of the peripheral nervous system are composed of Schwann cells and satellite cells. Okay, Schwann cells are larger cells of the peripheral nervous system, and they produce the myelin sheaths of axons. Okay, so they have a similar function of oligodendrocytes of the central nervous system. Okay, but here they're also present on unmyelinated axons. They're just not producing the myelin sheath around them. And they also have a function of repairing damaged axons of the peripheral nervous system. So there is some limited repair of nervous tissue. So here's a Schwann cell. Okay, here's a Schwann cell. So unlike oligodendrocytes that form the myelin sheath of the central nervous system, Schwann cells form the myelin sheath of the peripheral nervous system, but they only can form the myelin sheath on a single neuron. Oligodendrocytes are able to use their long arms to form the myelin sheath of multiple axons of neurons. Satellite cells are flat cells surrounding cell bodies of neurons in the peripheral nervous system. Okay, here they support cell bodies with processes linking to other parts of the neuron, satellite cells, and other Schwann cells. So here's a satellite cell. They're like little pieces of gum on the surface of the cell body. Okay, so finally, we mentioned that oligodendrocytes of the central nervous system and Schwann cells of the peripheral nervous system, their job is to form the myelin sheath. Okay. And now we'll talk more about the function of the myelin sheath. The myelin sheath is composed of the plasma membrane layers of the neuroglial cells. Okay, and its function is to insulate the electrical current to increase the conduction of action potentials. So those axons that are insulated by the myelin sheath will conduct action potentials faster than those axons that do not have a myelin sheath. Okay. Okay, so let's take a look more about the axons of the central nervous system and the peripheral nervous system. The axon segments that are covered by neuroglia are called internodes. Okay, the axon segments covered by neuroglia are called internodes. Between the internodes is a gap called the node of Ranvier with no myelin present, and these are present in all myelinated axons. There's also another area of unmyelinated part of the axon hillock called the initial segment. Okay, so let's go look at this. Okay, this figure is kind of tiny, but here we can see the myelin sheath. Okay, and the peripheral nervous system. And remember, we see the Schwann cell. Each Schwann cell is myelinating only one neuron. And you have areas where there's no myelin. That's called the area that's covered by the myelin sheath is called the internode. And the brief areas where there's no myelin is called the nodes of Ranvier. Okay. Now, if you look at the central nervous system, the oligodendrocytes, you can see the oligodendrocytes can myelinate multiple axons simultaneously. But they still have internodes where there's myelin sheath surrounding it. And areas where there's no myelin sheath are called the nodes of Ranvier. So both in the peripheral nervous system and the central nervous system, they both have internodes and nodes of Ranvier. Okay. Now, there's one other minor detail between the myelination between the two. In the peripheral nervous system, you can see that they wrap from the outside of the axon. Here, in the central nervous system, the myelin sheath wraps from the inside. We mentioned that unmyelinated axons of the peripheral nervous system are also associated with Schwann cells. Okay, but here the Schwann cells encloses the multiple axons more like a hot dog in a bun. Okay, so here's a Schwann cell, and even though they're not myelinating it, they're kind of keeping them protected, even though they're not quite insulating them completely. You can see that there's an opening for them.